but historically it actually was invented by chemists. In non-pharma industries, it's quite normal to be dealing with hundreds, if not thousands, of samples. For example, in the oil industry, in the mining industry, an example I'd give is a company prospecting for gold will take thousands of coarse samples from the soil, and they would assay the soil samples, and you would get a standard distribution of the concentration of gold ore or iron ore in the soil. Occasionally, though, you might have a sample which is pure gold, a gold ingot. That doesn't mean the whole mountain is made out of gold. It means you found a pocket of solid gold. So it's a different sample population from a statistical point of view. So in the initial test, that would be an outlier, but then you would separate it into a different subgroup of gold, solid gold. So the point I'm making there is, just because you get an outlier doesn't mean you want to reject the result. It's giving you information, and this is the whole philosophy behind outliers, even in the pharmaceutical industry. Is it's a good test to do to see if there's something very, very strange going on, or whether the sample is from a different sample population. Um, but it's not really of any use for rejecting results, so that you can pass a batch, which is where it's misused. Um, so for the pharmaceutical example, because you're expected to have thoroughly developed the product and have an operating envelope and develop critical process parameters and critical quality attributes, the idea that you'd get outliers is kind of causing problems for your validation. So here are the extracts from the original text on outlier testing, and just formally saying that if you've got a small sample set, then having an outlier is quite a delusion, because it relies on a standard distribution. And the pharmaceutical validation supports a standard distribution, otherwise a product wouldn't get validated. So this is kind of the loop you get into. The USP does allow it for biological assays, because biological assays for antibiotics and similar tests are well known to be variable, and biological assays behave in a manner all of their own. This slide just reiterates, in the pharmaceutical industry, you really can't use outliers for chemical tests using primary or secondary standards, because they're based on chemistry. There's no biological variation in there, and ultimately an outlier test cannot identify the cause of an extreme observation, and cannot be therefore used to invalidate a suspect result. There are several outlier tests. There's Dixon's one, and there's one called the Q test for sample sizes below 10. It's actually quite simple. You have your observations, and you put them in ascending order, and the outlier test is basically the difference between the farthest result, the one that you think is the outlier, and its next nearest result, whether it's at the high end of the group of numbers or the low end of the group of numbers. And you basically work out what's called the experimental Q value, and it's the ratio between the difference between the highest and the lowest and the range. In this example of the Q test, there are 5 results in ascending order. It's queried whether 56.23 is an outlier. The difference between 56.23 and the next nearest is 0.15%, and the range over the whole set of numbers is 0.28. The Q value works out at 0.54. So for 5 samples looking on the table, The 95% confidence is 0.73, which is greater than 0.54, so it's not classed as an outlier in this case. The R test, or the Dixon's test, is used more often, and it can be used for different sample sizes, so the table varies slightly as the sample size increases. So using the Dixon's test, you can see here for different sample sizes, the equation um, changes slightly depending on what range or what the differences you're trying to work out, but the use of the table is exactly the same. In this example, using the R test, again you find for the sample size of 4, the 95% confidence, the critical value is 0.765, which is greater than the calculated value of 0.55, so the result is again not an outlier. So here are some examples of the kind of citations you get for OSs, and if you read them you'll note that a few of them actually relate to not using your procedure properly. So the last one, releasing after OS using a grand average, including in spec and out of spec results. You can't average them. The prior one, not reviewing all the batch records properly the way you're supposed to. So it's not just having a out of spec SOP, it's making sure you follow it in exactly the sequence that you say you are. And to finish off OS, here are some of the findings and judgments of the original bar decision that happened in the mid-90s in America, and it started the whole subject off really because the company was found to be testing into compliance, averaging bad and good results, sampling and resampling, testing and retesting, no end in sight to how much sampling and testing you can do. The judgment was that you're not permitted to average out of spec results to get a pass. You're not permitted to conduct multiple retests without any predetermined limit. So you need to say if you're going to do 3, 5, 9 repeats. And also on the outlier testing, for chemical testing, you can't use it for like chemical assays. You can use it in biological or biotech assays, but not in chemistry, because your product is meant to be homogeneous and representative. So thank you for listening to this presentation, and if you have any questions regarding out of spec investigations or outlier testing, or indeed any other GMP GLP questions, just send me an email. I can also provide SOPs and forms and templates if you like. Hope you enjoyed it. Thank you.